Nucleic acid structure refers to the structure of nucleic acids such as DNA and RNA. Chemically speaking, DNA and RNA are very similar. Nucleic acid structure is often divided into 4 different levels: primary, secondary, tertiary and quaternary. Primary structure. Primary structure consists of a linear sequence of nucleotides that are linked together by phosphordiester bonds. It is this linear sequence of nucleotides that make up the primary structure of DNA or RNA. Nucleotides consist of 3 components: nitrogenous base, adenine, guanine, cytosine, thymine, uracil. 5-carbon sugar which is called deoxyribose and ribose. 1 or more phosphate groups. The nitrogen bases adenine and guanine are purine in structure and form a glycosidic bond between their 9' nitrogen and the 1' OH group of the deoxyribose. Cytosine, thymine and uracil are pyrimidines, hence the glycosidic bonds forms between their 1' nitrogen and the 1' OH of the deoxyribose. For both the purine and pyrimidine bases, the phosphate group forms a bond with the deoxyribose sugar through an ester bond between one of its negatively charged oxygen groups and the 5' OH of the sugar. The polarity in DNA and RNA is derived from the oxygen and nitrogen atoms in the backbone. Nucleic acids are formed when nucleotides come together through phosphordiester linkages between the 5' and 3' carbon atoms. A nucleic acid sequence is the order of nucleotides within a DNA or RNA molecule that is determined by a series of letters. Sequences are presented from the 5' to 3' end and determine the covalent structure of the entire molecule. Sequences can be complementary to another sequence in that the base on each position is complementary as well as in the reverse order. An example of a complementary sequence to AGCT is TCGA. DNA is double-stranded containing both a sense strand and an antisense strand. Therefore, the complementary sequence will be to the sense strand. Secondary structure. Secondary structure is the set of interactions between bases, that is, which parts of strands are bound to each other. In DNA double helix, the 2 strands of DNA are held together by hydrogen bonds. The nucleotides on one strand base pairs with the nucleotide on the other strand. The secondary structure is responsible for the shape that the nucleic acid assumes. The bases in the DNA are classified as purines and pyrimidines. The purines are adenine and guanine. Purines consist of a double ring structure, a 6-membered and a 5-membered ring containing nitrogen. The pyrimidines are cytosine and thymine. It has a single-ringed structure, a 6-membered ring containing nitrogen. A purine base always pairs with a pyrimidine base pairs with cytosine, C, and adenine, A, pairs with thymine or uracil. DNA's secondary structure is predominantly determined by base pairing of the 2 polynucleotide strands wrapped around each other to form a double helix. There is also a major groove and a minor groove on the double helix. The secondary structure of RNA consists of a single polynucleotide. Base pairing in RNA occurs when RNA folds between complementarity regions. Both single and double-stranded regions are often found in RNA molecules. The antiparallel strands form a helical shape. The 4 basic elements in the secondary structure of RNA are helices, loops, bulges, and junctions. Stem loop or hairpin loop is the most common element of RNA secondary structure. Stem loop is formed when the RNA chains fold back on themselves to form a double helical tract called the stem. The unpaired nucleotides form single-stranded region called the loop. Secondary structure of RNA can be predicted by experimental data on the secondary structure elements, helices, loops and bulges. Bulges and internal loops are formed by separation of the double helical tract on either one strand or on both strands by unpaired nucleotides. A tetraloop is a 4 base pairs hairpin RNA structure. There are 3 common families of tetraloop in ribosomal RNA, UNCG, GNRA, and QUNCG is the most stable tetraloop. Pseudoc knot is a RNA secondary structure first identified in turnip yellow mosaic virus. Pseudoc knots are formed when nucleotides from the hairpin loop pairs with a single-stranded region outside of the hairpin to form a helical segment. H-type fold pseudoc knots are best characterized. In H-type fold, nucleotides in the hairpin loop pairs with the bases outside the hairpin stem forming second stem and loop. This causes formation of pseudoc knots with two stems and two loops. Pseudoc knots are functional elements in RNA structure having diverse function and found in most classes of RNA. Dot-knot PW method is used for comparative pseudoc knots prediction. The main points in the dot-knot PW method is scoring the similarities found in stems, secondary elements and H-type pseudoc knots. Tertiary structure. Tertiary structure refers to the locations of the atoms in three-dimensional space, taking into consideration geometrical and steric constraints. It is a higher order than the secondary structure, in which large-scale folding in a linear polymer occurs and the entire chain is folded into a specific three-dimensional shape. There are four areas in which the structural forms of DNA can differ. Handedness, right or left, length of the helix turn, number of base pairs per turn, difference in size between the major and minor grooves. The tertiary arrangement of DNA's double helix in space includes bDNA, aDNA and zDNA. bDNA is the most common form of DNA in vivo and is a more narrow, elongated helix than aDNA. Its wide major groove makes it more accessible to proteins. On the other hand, it is a narrow minor groove. bDNA's favored conformations occur at high water concentrations. The hydration of the minor groove appears to favor bDNA. bDNA base pairs are nearly perpendicular to the helix axis. The sugar pucker which determines the shape of the A helix, whether the helix will exist in the A form or in the B form, occurs at the C2 endo. aDNA is the form of the molecule under dehydrating conditions. It is shorter and wider than helix B. Most RNA and RNA-DNA duplex are in this form. aDNA has a deep, narrow major groove which does not make it easily accessible to proteins. On the other hand, its wide, shallow minor groove makes it accessible to proteins but with lower information content than the major groove. Its favored conformation is at low water concentrations. aDNA's base pairs are tilted relative to the helix axis, and are displaced from the axis. The sugar pucker occurs at the C3 endo and in RNA2OH inhibits C2 endo conformation. Long considered little more than a laboratory artifice, aDNA is now known to have several biological functions. zDNA is a relatively rare left-handed double helix. Given the proper sequence and superhelical tension, it can be formed in vivo but its function is unclear. It has a more narrow, more elongated helix than A or B. zDNA's major groove is not really a groove, and it has a narrow minor groove. The most favored conformation occurs when there are high salt concentrations. There are some base substitutions but they require an alternating purine-pyrimidine sequence. The N2 amino of GH bonds to 5'PO, which explains the slow exchange of protons and the need for the G-purine. zDNA base pairs are nearly perpendicular to the helix axis. zDNA does not contain single base pairs but rather a GPC repeat with pp distances varying for GPC and CPG. On the GPC stack there is good base overlap, whereas on the CPG stack there is less overlap. zDNA's zigzag backbone is due to the C-sugar conformation compensating for G-glycosidic bond conformation. The conformation of G is in, C2 endo and for C it is anti, C3 endo. A linear DNA molecule having free ends can rotate, to adjust to changes of various dynamic processes in the cell, by changing how many times the two chains of its double helix twist around each other. Some DNA molecules are circular and are topologically constrained. A covalently closed, circular DNA is topologically constrained as the number of times the chains coiled around one another cannot change. This ccc-DNA can be supercoiled, which is the tertiary structure of DNA. Supercoiling is characterized by the linking number, twist and writhe. The linking number for circular DNA is defined as the number of times one strand would have to pass through the other strand to completely separate the two strands. The linking number for circular DNA can only be changed by breaking of a covalent bond in one of the two strands. Always an integer, The linking number of a cccDNA is the sum of two components, twists and writhes. Twists are the number of times the two strands of DNA are twisted around each other. Writhes are number of times the DNA helix crosses over itself. DNA in cells is negatively supercoiled and has the tendency to unwind. Hence the separation of strands is easier in negatively supercoiled DNA than in relaxed DNA. The two components of supercoiled DNA are solenoid and plectonemic. The plectonemic supercoil is found in prokaryotes, while the solenoidal supercoiling is mostly seen in eukaryotes. Quaternary structure. The quaternary structure of nucleic acids is similar to that of protein quaternary structure. Although some of the concepts are not exactly the same, the quaternary structure refers to a higher level of organization of nucleic acids. Moreover, it refers to interactions of the nucleic acids with other molecules. The most commonly seen form of higher-level organization of nucleic acids is seen in the form of chromatin, which leads to its interactions with the small proteins histones. Also, the quaternary structure refers to the interactions between separate RNA units in the ribosome or spliciosome. See also: nucleic acid double helix, nucleic acid structure determination, nucleic acid structure prediction, DNA nanotechnology, nucleic acid design, nucleic acid thermodynamics, cross-linking of DNA, non-helical models of DNA structure, DNA supercoil. References